Well, welcome everybody, and thank you for that introduction. So, yeah, I'm Sam. I'm a research scientist at Conifer Point, and today's webinar will be a continuation of webinar one. And today I will be discussing the development of docking methods, fragment maps, and how we will apply both to structure-based drug design. So as Jessica stated, for formatting of this meeting, I hope to discuss topics for about 45 minutes and then answer questions near the end. So yeah, please put those questions in the Zoom chat and we will address them then. So in episode one, I covered how to access PDB proteins and the 500 pre-computed proteins with over 100 simulated fragments that are available in BMaps. I also covered how to visualize and customize the view of proteins and the workflow associated with docking. So as that continuation of webinar one, today I will present the utility of BMaps as a tool for docking and lead optimization through the lens of factor 9, which is today's protein of interest, as well as a compound series that was reported on in the same paper. Specifically, we will take a deeper dive into evaluating the effectiveness of docking as a method of modeling and review common chemical characteristics found in the series today by evaluating docking poses and considering key chemical properties. Once the docking workflow has been reviewed, we will employ fragment-based methods and to generate novel designs and make decisions on the best next steps. This will include simulating waters and fragments, which will allow us to evaluate protein-ligand interactions and identify key residues for targeting with the goal of improving affinity. Okay, so at this point, I'm going to introduce 4YZU, which is a PDB entry crystal structure for factor 9, which is a vitamin K-dependent plasma protein involved in the coagulation pathway. The co-crystal ligand is a 9 micromolar hit that can be considered an early anticoagulant. So the left image shows the protein view of factor 9 in BMaps with the docking grid presented around the crystal ligand. The right image shows the crystal ligand structure, which has been energy minimized in gray, and the docking pose, which has been colored in blue here. Generally, when first exploring a protein and considering docking as a method for evaluation, I will dock the crystal ligand back into the binding site. In this case, I'm looking to make sure that the appropriate pose is found and that key interactions are replicated between the docking pose and the crystal structures pose. This gives me some confidence as I move forward with the evaluation of compound enzymes. And lack of that overlap raises some concerns, but may be traced back to the size of the docking grid or other docking parameters that can be adjusted. As you can see here, the docking pose provides strong overlap with the crystal pose and has a docking score of negative 8.8 kilocalories per mole, as well as an energy score of negative 67.2 kcals per mole. For reference, a larger negative score for either is an indicator of higher affinity. So while these scores are generally system dependent, these energy scores can be considered strong for a 9 micromolar hit. During this talk, I will predominantly consider energy scores, which are a BMAP-derived value that is based on electrostatic interactions, shape complementarity, and conformational stress. So the goal of this preliminary exercise is to dock a series of ligands with known affinity to understand what the limitations of the docking model are. In this way, we can proceed into novel chemistry with reasonable confidence. So in the 4YZU paper, the first table presents with various benzimidazole analogs as shown here. So they have been docked, minimized, and energy scores have been calculated. So the first value here is the Ki, which represents the concentration at which the inhibitor ligand occupies half of the receptor sites with no competing ligands present. This binding constant is presented in micromolar units alongside the computationally determined energy score and desolvation, which are in units of kilocalories per mole. So considerations for tautomers, desolvation, and other properties will be considered on further slides. Okay, so tautomers are a reasonable consideration in the context of modeling. So they're structural isomers, as shown here. These are each individually prepared and docked. And you can see that the hydrogen bond donor associated with the imidazole in both cases orients towards this blue 217. So, but due to the asymmetry of the groups coming off of this phenyl group, the positioning of the benzimidazole is shifted here. So this changes the key hydrogen bond's length as well as its geometry. And as you can see for these energy values associated with this hydrogen bond, it is reduced for tautomer 1. So for the scope of this exercise, higher affinity isomers are considered in the quantitative analysis. So we'll move forward looking at these structures. Okay, another key consideration is desolvation. Desolvation is another value that's calculated in BMaps, and it is the energy required to remove solvent from the ligand en route to binding. It's considered a detriment to binding, and therefore larger desolvation energies are undesirable. Compound 7G, for example, features this amidine, which is typically charged at physiological pH, and these charged structures tend to have a very large desolvation energy. So despite the addition of several hydrogen bonds, the combination of these factors will most likely result in a weak affinity, as we see here with this 54 micromolar Ki. So BMaps can calculate desolvation at the compound level, as we just looked at, but it can also highlight key regions and provide their contribution to the overall desolvation. Ideally, desolvation is low. For this metric, a functional group or regional desolvation under 3 kcals per mole is considered favorable, and anything over 10 kcals per mole is considered unfavorable. So as expected in the case of 7G, a significant portion of the desolvation is associated with the amidine. So this desolvation comes out at about 21 kcals per mole, which is comparable to the benefit of any of these additional hydrogen. So previously we looked at benzimidazole analogs. Now we can also look at the other side of the molecule for phenyltriazole modifications. Analogs on this side of the molecule feature fluoro and chloro modifications to the phenyl, as well as variations to the triazole configuration. Additionally, there's imidazole that's been presented here. So upfront, some qualitative observations can be made. For example, this 2 position on the phenyl, the chloro has the strongest affinity, followed by the fluoro, and then the hydrogen here. So when we look at docking poses, we'll see that this position is solvent exposed, and so the more polar groups here are going to be more preferred. Additionally, for both qualitative and quantitative analysis, it's really critical to have appropriate poses. So the example here is the 7P structure, where during an initial screen, we ended up with a pose flip. So it fills the space appropriately, but this benzimidazole is residing in the place that typically we see these phenyltriazoles. So this isn't necessarily appropriate for comparing energies within a congeneric series. And so for this reason, a subsequent screen was applied with an increase in pose outputs, and we were able to find the appropriate pose shown here. So this resulted in a significantly stronger energy score that we will see later is much more representative of its affinity. Okay, let's see. So 7R shown here presents with this 1,2,3 triazole, and it has an inappropriately strong energy score compared with its binding cuts. So the reason for this issue may be unclear in a general docking screen. 4YZU does come with crystal waters, but understanding the energetic properties of waters in a variety of positions is important to understanding the bindings. Therefore, water maps were calculated through BMaps, and a simulated water was found at the bottom of the binding. So this suggests a much more polar region than is observed when modeling without waters, or only with the crystal waters that come with the PDB structures. So partial charges are shown for the highlighted pink atoms, as well as for the uppermost proton on this simulated water. You can see that the partial charges are consistent for the water, partially positive. And then for compound 1, this pink highlighted water has a negative partial charge, negative point. Compare this to the partial positive charge for this carbon atom highlighted for 7R at 0.27. So we can see that there is some charge complementarity here, not necessarily a hydrogen bond that's explicitly shown, but for this reason, we would expect to see compound 1 having a better affinity, even though the modeling didn't necessarily capture this. Additionally, so this water that we found does provide for an additional target for creating hydrogen bonds, or potentially we could explore the idea of displacing this water and forming a hydrogen bond to this 3-175. So at this point, I'd like to take a moment and discuss the water maps that are simulated in BMaps. In certain circumstances, docking does not return poses that are consistent with the crystal structure ligands. One reason for this may be that key waters exist in the binding site that are involved in water-mediated hydrogen bonds, or as we've just seen, those waters can alter the electronic geography of the region. So the output for BMaps waters can allow a user to determine a ligand's ability to dislodge or preserve that water, as well as say what next steps you would take. So these pink waters, as well as these gray waters, are considered tightly bound waters with higher energy, whereas these green waters are weakly bound waters that are ideal targets for displacement. So in practice, weakly bound waters can be easily displaced, and there's an entropic benefit for displacing waters. But tightly bound waters can also incur an enthalpic penalty. The modification provides a sufficiently strong interaction, and displacement of these stronger waters can be energetically favorable and lead to additional activity. So there's a final position on the molecule that was analyzed in the paper, and that's this ethylene linker group. So chiral groups were presented from both carbons here. Chiral phenyl group was found in 7W. That leads to a very low nanomolar affinity. It's important to note that these compounds carry the 2-fluorophenyl that was observed in 7O. So these modifications, these analogs had 2 modifications compared with the original compound. All right, so taking a look at 7W in a little bit more detail, there's a lot of information on this slide, so I'm gonna go right to left on this. First, taking a look at this ligand interaction diagram. First, we can see that we have captured the original hydrogen bonds from the crystal structure in blue 217 and glide 216. So these are presented. Additionally, we pick up a couple of hydrophilic interactions here, I guess hydrophobic interactions off of this fat. So the lower central image is shown in pink, 7W is shown in pink, and in gray, this is that original crystal structure confirmation. We can see here that they have a high degree of overlap and should be considered relatively reasonable for analysis. And finally, in this left image, what I wanted to show is that arginine 143 is shown to have two occupancy states, shown in red and in purple. This indicates to an extent that this is a highly flexible residue. But as of now, there are no clear interactions observed between this the side chain and the new phen. Right, so a couple of hydrophobic contacts is not likely to give us this 16x increase in affinity from the 7 O's to 60 nanomolar to the 7W 16 nanomolar. Right, so for this reason, it's hypothesized that there's an energetically favorable cation-pi interaction formed between this arginine's side chain and the newly added phenyl group. Next steps to catch this hypothesis, or to chase it down, I suppose, would be to involve simulations that provide full protein flexibility, but the nature of this interaction is not explored further in this analysis. I'd also like to discuss some of the other properties that are calculated in BMaps that have come in handy in various projects. So first is C log P, which is a predicted value for the log of the partition coefficient. Log P is considered an indicator of molecule density. Within Lipinski's rule of 5, a desirable log P should be between 0 and 5, with a larger value indicating a compound is more lipophilic. In this case, there's a weak relationship between affinity and log P, indicating that larger C log P corresponds with higher affinity compounds. Second, we have polar surface area, which is commonly considered as a property in drug design, and we can take a look at that. So within this congeneric series that we're analyzing, many comparable compounds that present with the same polar surface area. So in general, we can say that there really isn't much of a relationship between polar surface area and our affinity. All right. So here I've plotted energy scores for the initial docking of all the compounds against the log of the binding constant. As I stated earlier, only the higher scoring tautomers were considered. And what we find from this is there isn't really a notable relationship with energy score and assay data. But we can apply everything that I just talked about in order to appropriately curate this data. So here are two key actions we're taking. First, the flipped pose that we discussed associated with 7P can be replaced with the correct pose. And this provides a significantly more agreeable data point that is comparable with the others. Second, the amidinyl 7G was flagged due to its undesirable desolvation value. This structure and its energy score can be excluded as long as any compounds considered down the line are also valued in the context of desolvation, and those that are similarly flagged are evaluated in some other way. So by curating this set and establishing a standard for expectations moving forward, a correlation between the computationally obtained energy score and affinity is obtained with an R squared at 0.55. So in the end, the data trends as expected, with strong affinity compounds receiving stronger energy scores, and weaker affinity ligands receiving weaker scores. All right. So as we transition from validating and reviewing existing data to evaluating new compounds, it's practical to locate fragment hotspots. This can be used to find regions to extend a ligand or to identify potential allosteric sites. It's important to note that the hotspots shown here are part of the pre-computed data that is offered in BMaps. So the ligand view shown in the central image highlights 3 key hotspots, numbered 2, 6, and 7, and provides the tooltip for hotspot 2. So it shows that imidazole is a key fragment that has a close relationship to the 1,2,4 triazole that we've been previously looking at. Additionally, an acetamide might be a desirable group, and we'll explore that down the road. So hotspot 2 is highlighted largely because it has this target water presented at the base of it. And then diving a little bit deeper into this hotspot, it shows significant space below the triazole. And then the tooltips shown here are for the energy of this water, as well as the energy for the hydrogen bond between the water and threonine 75. So this water represents a reasonable target for either a water-mediated hydrogen bond or as a water to be displaced. So as we move forward, that's kind of the goal that we're trying to keep in. And so now I've established a goal for modification. And before I discuss the results of fragment screening, I'd like to take a moment to discuss how our fragment simulations work. So at the earliest stage of the simulation, the protein is placed in a simulation box big enough to accommodate 3 layers of the desired fragment. In the case shown, this is 1. So the simulation is started with a high chemical potential, where fragment molecules rapidly fill the box and occupy all cavities at the surface and deep within the protein. As the chemical potential is lowered, not much happens until this free energy reaches a critical level that is capable of overcoming solvent cohesion, and the system goes through a phase transition that evacuates the bulk solvent and most of the protein-bound solvent, exposing high affinity sites. Continuing to lower chemical potential further reveals fragments with extremely high affinity. So this plot is of the number of fragments versus the chemical potential, and it shows the abrupt transition when the bulk solvent is evaporated out of the site. So the fragments that result from this simulation and their positions can then be reviewed to determine if they are suitable for ligand modification. All right, so following a fragment simulation, while applying over 100 different fragments, as well as a fragment grow vector shown in blue, was selected, extending away from the phenyl and into hotspot region 2. The top 10 fragment results are captured in this table, and those show the binding score here. Oops. The binding score, as well as proposed linker types that would be most beneficial for connecting the fragment to the determined connection point. So an example of some of the variation here is on this 2-pyrimidone, which presents in the 2nd and 3rd positions on this table and has both a direct connection as well as a methylene connection. So it's important to note that some of these linkers are methylene and ethylene linkers that extend farther into the pocket and will allow for further interrogation of the region that is containing the target water. All right, so the appended compounds were docked and evaluated for their energy scores. So it's important to note that all of these compounds have an aromatic group as well as are accompanied by various heteroatoms and halogens. Notably, all of these compounds have significantly improved energy scores compared to compound 1. And this highlighted compound 8 has an improved energy score compared with 7W, which was the 16 nanomolar hit. So analog 8 is shown here in 3D. We can see that it retains the hydrogen bonds to glycine 216 and as well as 17. Additionally, it has a hydrogen bond interaction to this target threonine 175, and even has a higher hydrogen bond energy than we saw for the water. Doing all of this and also maintaining optimal. All right, so including the high scoring analog 8 that we developed, I've also chosen to review the phenylbromide 7 and a comparable phenol that kind of bridges these two structures. So bromide analog 7 does not present with a hydrogen bond donor. Therefore, it doesn't benefit from the hydrogen bond that we observed with 8, as well as this desmethyl N. Comparatively, the phenyl hydroxyls do interact with threonine 75. So the methyl analog presents this methyl towards solvent and also has a hydrogen bond of negative 8.8, whereas the desmethyl analog has a stronger hydrogen bond at negative 9.1 and is also allowed a little bit more free rotation within this pocket. So it's important to note here that arylbromides and phenols tend to have metabolic issues. The arylbromides are metabolized in the liver, and phenols are typically a concern is CYP substrate. So with this in mind, we're going to take a look at some other potential analogs that might not present with similar issues. So because those other compounds, the previous ones, might have some ADME issues down the road, these were some other analogs that considered. So this N-acetyl-12 was obtained through a secondary fragment screen, and compounds 13 and 14 were inspired by the hotspot analysis, which suggested that acetamides would be potentially beneficial in this region. So you can see that compound 13 presents with a hydrogen bond to that target threonine 175. Additionally, it fills this pocket quite nicely and might make for a good compound. So through the work discussed here, a series of new compounds were modeled and predicted to have strong affinity. You might, after, let's say, a benchtop screening of these analogs of compound 1, additionally add that chiral phenyl to the ethylene, as well as the chloro to the phenyl here, in order to further improve affinity. And then to circle back to earlier slides, it's important to note that these modifications do not appreciably increase desaltination. And also, while there is an increase in polar surface area for these two acetamides, they're all within the bounds of reasonable polar surface area. So this appears to be a very good series of compounds to move forward with into synthesis and testing. So to summarize today's discussion, first, ligands with a ligands with a known affinity were leveraged to establish a workflow and understand the protein and ligand system of interest. Afterwards, a variety of analogs were developed based on fragment-based approach. And then standout ligands with ethylene-linked phenols were explored further, and additional modifications were proposed. And then finally, chemical properties and key energy scores were reviewed to ensure the best selections. So today was an example project explored BMaps, that was employed docking, fragment-based analysis, as well as water simulations, and showed hotspot analysis. So all of this together highlights BMaps as a powerful tool to develop new compounds with ideal properties. So I got there a little fast, but at this point, I'd love to take some questions and perhaps chat about what we talked about here.
Thank you so much. Now, everyone, if you want to post any questions in the chat, feel free to do so and we'll read them out. Sam, I have a question for you regarding the prioritization of fragments versus the waters that we're seeing we'd like to replace. How is it that you think about which fragments will be able to replace the waters that we find using our water maps?
Mm-hmm. Well, right. So we have a couple of considerations with this, and it starts with the strength of that water. So if we're kind of balancing replacing a water, we need to have a fragment that's of higher affinity. There is that sort of entropic benefit that goes along with displacing that water. But if we're not doing so with a stronger affinity than the water itself, then it's not going to happen. So number one, we're looking for strong affinity fragments. Number two, considerations for what is that water doing in that binding pocket? Is it involved in hydrogen bonds? Is it linking residues? And then we're either looking to mimic those similar interactions or find other interactions in their region.
Excellent. So one question I got is related to fragment sets. You demonstrate how you can make fragment maps or you can make water maps, but we can also make fragment maps in Boltzmann maps. Can you talk about the different fragment sets that are already available in BMaps and whether or not we can add our own?
Absolutely. So to begin with, there are several series of fragments that are currently available. So that includes what we call our favorites, which involves a lot of very common for fragments. And then additionally, you can create your own. Those can come through a couple of different avenues. Number one, what I find myself doing the most is taking groups that would be consistent with the original hit, and then adding new functionalities to that. That is to say, if we're looking to replace a phenyl, we might add a methyl to that, or other heteroatoms that might be meaningful in the context of the protein, and then modeling those. So you can create your own list and then import those in. And then also we have a decomposition tool, which allows you to take ligands perhaps from a library that you have and decompose those into fragments and then run them back through.
Okay, fantastic. So you don't necessarily have to have a fragment set yourself. You can just take compounds and break those up into fragments.
Absolutely.
Awesome. We have another question in chat here. I have a general question. In the case of GPCRs, how much water map scoring with BMaps are successful? Because GPCRs are membrane proteins, and by their nature, they are hydrophobic. They are in a hydrophobic membrane environment. So what experience have you seen with GPCRs and our water maps, Sam?
Well, I have some experience with membrane sort of membrane-based proteins. And I can say that, yes, it does become more difficult, right? You're balancing that, the polar surface area, permeability, and what I found is that, there's a heavy reliance on some of these other properties as you move forward. Something that has a higher C log P might be valuable in order to achieve better permeability. And so these are the kinds of things that, like I talked about today, you kind of validate each system on its own and then move forward with new modifications. So looking at those properties is a great way to start considering what will actually make it to your target mining site, even if it's sort of internal to the membrane.
One point that our CEO, John Culp, wanted me to point out is we actually have a water map benchmark paper going out, or that was submitted this month. And it shows that BMaps is the most predictive commercial tool for water maps. And actually, there's an example where you can do water maps of a protein membrane system. So look out for that paper to see more about membranes. Another question that was asked is, in terms of computational costs, how long does it typically take to generate water maps and hotspots?
Yeah. Well, it depends, number one, on the size of protein. Larger protein tends to take longer to simulate, but we've done a lot in terms of optimizing. So you can, within BMaps, establish a box where you want your fragments to run and eventually converge in. And that's reduced the time for fragment maps considerably. To that end, you can, I want to say it's probably a few hours for a fragment screen, but waters tend to take a little bit longer. It also depends on the fragments. A more polar fragment is going to end up taking longer as you ramp that chemical potential. So you could expect to have a pretty respectable set of fragments by tomorrow if you were working right now.
So generally hours to a day, depending on how large your fragment set is and how large your protein is. We have a question. Will there be another webinar?
I think that we probably should have more that involve discussing what those fragment sets are and continuing to dive into how we could use fragments. Additionally, I think as new features come into BMaps, I think that would be really exciting to kind of show what we can do.
Absolutely. And we, of course, would love your feedback. Anyone in the audience, if there's a specific webinar topic you would like us to create a webinar around, please let us know. There's a question, what is the recommended hardware requirement? HPC, GPU? That's an excellent question. BMaps is a browser-based software, so it will run locally on your client machine. If you are looking to run fragment maps, we have the ability to submit those directly to AWS. That's already set up to use Spot instances, so it's a very quick and low cost methodology there. But in terms of running Boltzmann maps to visualize your systems, to look at the over 500 different proteins that we already have populated with fragment maps using our entire fragment set, that's all available right now in your browser. And one of the favorite things that I like to show off is you can run Boltzmann maps from your phone. So if you want to bring up one of your sessions that looks at a protein, especially if you're at a poster session and you don't want to be stuck in the framework of whatever picture 2D image that you made on your poster, you can pull that protein and their respective compounds up in Boltzmann maps and spin around the protein in a full 3D diagram to show off your science right there using just your phone. So it's very lightweight application. You do not need an HPC or GPU to use it. Okay, with that, I will thank everyone here in the audience. Thank you, Sam, for a wonderful webinar. And if you have any questions, please do send us a message using support@coniferpoint.com. At Boltzmann Maps, we are constantly trying to make our software and our available features more useful for our users. So definitely let us know if you have any recommendations or if you have any questions on how to get started with BMaps.